So I may refer back to that kind of stuff now. The both of those I made up before I had this book. The previous book that I used, Fox, which is a fantastic book and is actually much thicker than this. In some ways it's a little bit more advanced. I still like this book better. Did not have a lot of this in a very organized fashion, so I generated these things. Okay? This book does have them, shockingly enough, because one of the co-authors of this book was one of my professors at the medical college. It's the way I learned how to think. Shocking, he writes a book. The sections he puts in there look a lot like the way I think. So that's why there may be some redundancy with the two. We're going to work with mine just to stay consistent. But the stuff that's in the chapter is the same. Okay, mine are a little bit more detailed. But the idea is the same. So that's something, again, you're going to have to learn. Okay. Here we go. Yes.
So really quick from last
Time.
lecture, yeah.
Yep.
So when talking about muscle stretch, looking at this.
Yeah. Golgi tendon, organ, and muscle spindles. Yeah.
Why is it that this has an alpha neuron when this wouldn't have a here? I just don't understand, like, why is this? If these two are pairs when they're doing.
Why is this
So I
not
don't-
activated?
Why does this not like gamma?
So this is
What's this?
this is the alpha. This is the one. So what this is saying right now is that there's nothing going on. All right. Now that we've seen Ryan, good question. All right, muscle. So the first and most important thing is something called the sliding filament theory. Okay? So there are two major proteins that make up the sarcomere. The sarcomere is the major functional unit. I like to call them functional units. And it's the smallest functional unit. The sarcomere, this is garbage too. Is the functional unit of muscle. Now, it really only applies to skeletal muscle and cardiac. Smooth muscle is a slightly different beast. Okay? It's the functional unit. Of that, and when we draw a sarcomere, I'm going to draw it very simplistically. It's much more complicated than this. Okay? This is a very simplistic diagram. These are called actin fibers. Okay, and this big honkin' one here, which I haven't drawn, is called myosin. The myosin is called a thick fiber. and this is called the thin fiber. Okay? So that's a really simple form. They're going to show you much more complicated. Sarcomere. And if you line a bunch of these things up, then you ultimately will make fibrils, and fibrils will then become fibers. Okay, sliding filament theory. What does that say? It says that when you have a muscle contraction, the thick and the thin filaments will slide by one another. Okay? They will slide. Actually, the thick filament doesn't move. Only the thin filaments will move towards the center. So that's called a sliding thick filament theory. So you can imagine that this is the sarcomere, and as it contracts the muscle, it gets shorter. The filaments will slide by one another until they get as close as they can possibly get. But they don't actually touch, right? This doesn't physically touch this. Part of this will touch this. That's not good either. We're going to talk about these heads. The heads that are attached to myosin, the myosin heads actually touch. But this part of the thick filament does not touch. It's the heads. All right, so we'll talk about that more. Sliding filament theory. As it gets smaller, they slide past one another. In all three types of muscles, myosin and actin interaction are regulated by the availability of calcium. The reason I didn't put smooth muscle in here is because smooth muscle is slightly different in its characteristic. It has myosin and actin, but it doesn't look like this sarcomere here. It's a very different configuration. Okay. So calcium becomes very important. In the human body or in any physiological system, anything that contracts or secretes something has to involve calcium. It is involved. So that's no different. So in all three muscle types, the interactions between actin and myosin are regulated by calcium. The more calcium you have, the better. Okay? Changes in the membrane potential of muscles are linked to internal changes in calcium release. So this you begin to understand now because you've seen this neuromuscular junction thing, which I'm going to refer to. You know what a membrane potential is, right? The change, the potential energy. The muscle, just like a neuron, has a membrane potential, right? There's really only two excitable cells in the body in its simplest form, a neuron and a muscle cell. It's not 100% true anymore because now we have GI cells and some glial cells that actually respond. But for your intensive purposes, the neuron and the muscle fiber can both be stimulated to do something. They're excitable cells. So this works much like a neuron. Neuronal influences, again, you can write this stuff down. Nothing here that you haven't heard before. In smooth muscles surround tubes, right? So smooth muscles surround your stomach, 3 different layers in 3 different directions. Smooth muscle surrounds arteries and veins. Smooth muscle surrounds your intestines. It's a very different type of muscle. Smooth muscle tends to be high endurance. Not like running high endurance. Not like even running a marathon high endurance. More endurance than that. Okay? So these muscles have more endurance than whatever skeletal muscle you have that's sort of designed for longer activity. It goes on for hours and hours and hours and days and days and days and days. Your skeletal muscle can't do that very easily. Right? So it's a much higher level. So that's what this says here. Okay, so you're going to have to know the breakdown as we go from a muscle belly here. So this is in the biceps, right? You have the full muscle, and we go down from the full muscle. The full muscle is surrounded by connective tissue. All right. So I'm going to draw this table. It's a very simple table. And this is, I like to call this the hierarchy of muscle and connective tissue. So on this side we have the muscle tissue. And on this side we have the connective tissue. So we're going to go from largest to smallest. Everybody with me so far? Okay. So in its largest sense, you have a whole muscle. Pick one. Example, bicep. Okay? So you have that. Then the next level down is what's called a fascicle. Then the next level down is called a muscle cell slash muscle fiber. Muscle fiber and muscle cell means the same thing. The reason some people call it a muscle fiber is because it looks like a fiber. It's long, but it's really a cell, right? And so one of the terms you may hear or see is muscle fiber, muscle cell. You have to know that they mean exactly the same thing. They're not any different. It can also be called a third term. It can be called a myofiber. So, a couple of things. Whenever you see sarco or myo, they're always referring to muscle. All right? Sarco anything, sarcopenia, sarcoplasmic reticulum, myofiber, myofibril. They're always referring to muscle of some sort. Okay, so these three mean the same thing. Then, as you go down, now you have what are called myofibrils. Now you can see how this starts to get confusing, right? Because a myofiber and a myofibril sound a lot alike. Right? They're even spelled almost alike, but they're not. They're very, very different. This is a muscle cell. This is an organelle. A myofibril is an organelle. Just like the endoplasmic reticulum or the nucleus or a ribosome. Okay. The myofibril is then made up of sarcomeres. Remember, the sarcomeres are the functional unit of muscle. Okay? The sarcomeres are then further broken down into myosin and actin, which are called, myosin and actin are called, here we go, myofilaments. You see how this can start to get really confusing? Myofiber, myofibril, myofilament. It's all MF. Okay? So from largest. Now, when we're talking about connective tissue, right, we're talking about the connective tissue on the outside here, you're talking about what's called the epimysium, associated with whole muscle. Then with the fascicle, you have something called a perimysium. And then with the muscle cell, you have an endomysium. The connective tissue surrounds the part of the muscle that I'm pointing to with the arrow. So the epimysium surrounds the whole muscle. The perimysium surrounds the fascicle, which is the next layer in between. And then the endomysium surrounds the muscle cell. Okay? Now, if you want to go bigger than this, then you have something above this, which isn't necessarily just connected to muscle, right? And it's called fascia. And actually, the epimysium is a part of the fascia. Fascia, if you can imagine yourself having another layer underneath your skin of like saran wrap, it's clear, and it connects all of the parts together, right? That's what fascia is. And part of the fascia over the muscle is the epimysium. But the fascia connects the bones and the tendons and the ligaments and the organs and everything together. So this would be broadest, but this isn't specific. Fascia isn't specific to the whole muscle. The epimysium is specific to the whole muscle. Okay, so that's something you should know. I don't know if they do that with the book. It's a table that I came up with many years ago, and I think it very nicely sort of puts everything together in terms of your understanding. Okay, so you should know the hierarchy, and I call this the hierarchy of muscle and connective tissue. You should go down, right? So here's a muscle fiber, here's an organelle, right? Here's a myofibril, and this part, when you take a bunch of sarcomeres and you put them end to end, you make up a myofibril. That's what a myofibril is. It's a bunch of these sarcomeres put to end to end. So if I were to draw this, right, what would it look like? Well, with my fantastic art ability here, right, it would look something like this. Right, and they're linked. So this would make up a myofibril. So when one of these contracts, they all contract. Right? You can't just contract this one or just this one. You can't do it. Right? If one of them contracts, they all have to contract. So that's what they're showing you here. Now, what else do you need to know about the sarcomere? The sarcomere is broken down into subparts. You also need to know this, right? So you start with the middle. The middle is called the M line. You can think about it as the middle. M for middle. It's not really what it stands for. It doesn't matter what it stands for. M line. That's all we need to know in this class. M line. Okay? Right down the middle of the sucker. Then, on the ends, you have these Z lines, right? And they look like this in a Z formation. So, one sarcomere is between two Z lines, right? Z line to Z line, that's one sarcomere with an M line in the middle. Are you with me so far? You can then further break down this sarcomere into what's called the H zone. The H zone is an area here. These are myosin, the thick filament, these brown pieces here, and the area around the middle that only has myosin in it is called the H zone. Only has, right? And you can see it here. There's only myosin here. There's no actin because the actin's to the right or to the left. Only myosin in here. The M line is the middle of the H zone. Does everybody see that? M line is the middle of the H zone. It's also the middle of the sarcomere. Now this is the one that gets somewhat confusing. Okay? Well, there's a couple of them that get confusing. So let's start with the I band. And the reason that the I band gets confusing is because the I band is where there is only actin. Actin, 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 actin. There's no myosin in there. But this, in one sarcomere on one side, you only have half of an I band. Where is the other half? It's on the other sarcomere. So it takes two sarcomeres to make a complete I band. And in that area, you only have actin. And that's what they're showing you here, right? They're showing you that here's the sarcomere, and that the I band floats over both of those. So that's the only one that you, in order to get a complete part, you need to have at least two sarcomeres. If you wanted two full I bands, you would actually have to have three sarcomeres that were connected, right? You'd have one here and one here. If you only have one sarcomere, you don't have a complete I band on either side. And then the A band. Now the A band is a little confusing too. The A band is any area that includes actin and myosin together. So if you look at that, it's really wherever there's brown and purple, so actin, myosin, actin, myosin, actin, myosin, actin, myosin, which also includes the H zone. Okay? Which also includes the H zone. So most of the A band is made up of all of the areas where the actin and the myosin overlap, including the H zone where there is only myosin. That's a caveat. You have to understand that, because what I have people do on exams, and I've seen it before, right, is I say, draw me an A band, right? And they look at this and they say, well, it's only where the two overlap, right? So they do this. Because technically here and here, this is where there's actin, myosin, actin, myosin, actin, right? So this is what they write. They write A band, A band. That's wrong. The A band stems from here all the way to here, including the middle of the H zone, which doesn't have any actin in it. Why am I saying this? Because this happens every year. And I say it every year, but I've never said it quite like this before. So this is a first. Right? I want to make sure that you don't make that mistake, because I've actually seen this mistake occur on an exam. And they say, but this is A band and this is A band, and I say, yeah, it's sort of right, but it's not correct. It's not the best answer, because the A band goes from one side to the next. Okay? So does that make sense to everybody? I don't want you to get it wrong, because that's just a foolish mistake to make. You don't need to lose points on that. All right. Then what we do here, what we have here is we've got this myosin, this thick filament, right, is broken down. All right, so they've broken it down, and this is the thin filament, and this is the thick filament. All right, myosin has these double golf club heads on it. That's what I like to think about them as, right? So you think of that, this is a golf club, and there are two golf clubs coming out, and they come out like this in a Y formation. And the golf, what goes in between the two golf clubs? What goes between the two golf clubs? It's hard to tell because this isn't 3D. But if it was 3D, you'd be able to tell. What do you think goes in between the two golf clubs? The actin, that's right. So in order to get a contraction, right, here's the actin, here's the golf clubs, and when you're not contracting the muscle, those golf club heads do not touch the actin. We're going to talk about how that happens. As soon as they touch, they clamp on, a contraction occurs. And then in order for your muscle contracting to stop, those two golf club heads have to come off. And everything goes back to the way it was. And so it's not just one golf club doing this, right? It's two golf clubs doing this and ratcheting. Right? So that's the thick filament. And I want to hold on. Let's see. Bear with me a second. Oh yeah, I want to get to this. I'll come back. The reason I want to talk, because this is what I want to talk about. So if you break down those golf club heads, I wish they actually had this in that back slide. When you break down those golf club heads, what do they look like? Well, the actual handle of the golf club are two heavy chains. So the handle, the long part here, is called the heavy chain of the myosin complex. The two actual heads, and they're called globular heads, are called the light chains. The light chains, the heavy chains. Together they make up myosin. On the light chains, you have two knobs here. You have a knob here and a knob here. Okay? And we'll talk about what all these knobs do. The actin, the purple knobs here are what physically bind to the actin. All right, so these two purple sites will bind actin. The brown spots here will bind ATP. That's where the energy molecule comes from. And in muscle contraction, the ATP, at least at this level, gets used to actually disassociate these globular heads from the actin. It's not actually used in the contraction. Think about it like a gun. If anybody's ever seen a pistol, right? An old, like a revolver, where you actually have to cock the head. You ever seen the old westerns where they've got to pull that back? Once that the hammer is cocked, all you have to do is press, right? Is pull the trigger, and boom. You're actually using energy. Most of the energy you're using is to actually cock the head, right? Same thing that happens to skeletal muscle. You're actually using energy to separate the two and to recock the head. Once the head is cocked, it's pretty easy to pull it. Like if you've ever shot a gun, a high-powered rifle, a semi-automatic or an automatic, they're touch sensitive, right? It doesn't really take a lot of pressure to fire a lot of bullets in these things because of the mechanisms and the way they're cocked. A revolver's not quite like that, but all right. Anyway, so and then you have these actual light chains. Now these light chains in the skeletal muscle don't do very much, but when we get to smooth muscle, they actually do a lot. Okay? So we'll talk about those. So any questions about this? I wanted to show you this, and then we'll go back. Okay. Any questions? Because we're going real deep into the cell biology here, right? So then you have the actin, and the actin here is made up of the actin here. All right? So you have different parts to the actin. So you actually have these beads here, these pearls, these blueberries, call them whatever you want. These are called G-actin. Right? So every one of these marbles is called a G-actin molecule. When you take a bunch of G-actin molecules and you put them together in a chain, a string of pearls, it is now called F-actin. So what we're actually, when we refer to actin, what we're actually referring to is the F-actin, right? Which is a combination of all these G-actin molecules put one after the other. So that's the marbles. And notice how there's this twisty shape, kind of like a DNA, kind of like a double helix look to them, right? Doesn't have like a DNA double helix type look. You then have this blue string, which also has this DNA double helix look. The blue string is actually called tropomyosin. Okay, it's hard to see in this picture. This is not a very good one, but I'll show you a better one. But on these beads, on these G-actin molecules, there are these dark spots. Those dark spots are myosin binding sites. Okay? So what does the tropomyosin do? Tropomyosin covers up the active sites on the actin beads. So these blue strings sit over an active site. If those blue strings do not exist, if they do not sit over that active site, you will be in perpetual muscle contraction. You will not be able to uncontract the muscle or relax it. So these blue strings here prevent that from happening. When we get to smooth muscle, they don't do that at all. They're a remnant. They're there for structural purposes. But in skeletal muscle and cardiac muscle, the tropomyosin prevents the ability of the myosin globular head to bind to its binding site on a G-actin molecule. That's what it does. Okay. Then attached to both the actin and the myosin are these three jelly beans in red here. Okay, those are called troponin. There are three parts to the troponin molecule. All right, so there's troponin T. Troponin T binds to tropomyosin. There's troponin A. Troponin A binds to actin. Now they're all bound to one another, right? So they're already glued to one another, the three of them. And then troponin I binds to calcium. And usually we think of the middle one here as troponin I. It's another receptor, you can think of it as. So those are all the parts. So you have to, so there's a lot of memorization here straight off the bat with terminology and stuff you need to know so that you can understand how we can manipulate the system and how it works. So right off the bat, one important thing, always you got to have memorized sooner than later, right? Because if you don't, then everything I say will be like, well, what's he talking about? I don't know what he's talking about. Either that or you're going to have your book open all the time. Better sooner than later. So take your way all the way down. Start with the hierarchy of muscle and connective tissue, right? Work your way down and backwards so that you can do it without, how do you know? Write the whole thing without a book. If you can't do it, you don't know it. It's as simple as that. If you can't do it every time, you don't know it. That's how you know. Are you ready for the test? Can't do this? Not ready for the test. Right? Test yourself. In this class, what you need to do for all of the topics to know if you're ready is test yourself. If you think you're ready, prove it to yourself. I think I would. That's not good enough. Prove it to yourself. You've got to have a way of testing. Okay. Here's the thing blowing up. Right? The one thing we haven't talked about yet, so here's, right, we have all the different zones, you have myosin and actin, are these spring-like structures. Now, these spring-like structures go all the way from a Z line, or Z disc, to the M line. You can only see part of it, but it goes all the way through, and it's called titin. Titin. Titin is the biggest protein in the human body. There is no protein bigger. And what does titin do? Well, titin is like a spring. So when your muscle contracts, what do you do to the spring? When the muscle contracts, when this end and this end come closer together, what happens to that spring? What happens to the spring when they come together? What are you doing physically doing to the spring? What? You're compressing the spring. Okay. Compressing the spring. Your cars have big old springs on them, one for each tire. Go underneath and look at it. There's a big spring. Okay? What happens when you hit a bump? Right? You hit a bump. What do you do to those springs? Compression. What does the spring do back? It uncompresses. Right? So that's part of your suspension system. So when you're actively contracting, you're compressing the spring. When you relax and the globular heads come off of the actin, what does the spring do to the sarcomere? It decompresses. There you go. Go back to the starting point. That's what titin does. It's also structural, right? It adds strength and it's a connector. So it has a function in terms of what it physically does, but it also is structural, meaning it holds this whole kit caboodle together. It's part of the physical structure. If you got rid of the titin, you wouldn't have as much of a connection to the Z line. All you would have is the actin. Right? The problem is that the myosin would be floating in space. And that would be bad. Because then it wouldn't be lined up. So it makes sure that everything stays lined up and that everything stays together. So it's also a connective protein. Right? Questions? Questions? All right. So if you take a cross section of muscle, meaning I take my bicep and I cut it this way, right? I take a saw or a scalpel and I cut it this way. And now I'm looking at it in this way. So that's what we're looking at. You can see the combinations of sarcomeres and, excuse me, not sarcomeres, combination of myosin and actin. So each myosin can interact with 6 actin filaments, right? Well, each myosin, the big brown dot, can connect to 1, 2, 3, 4, 5, 6. That's where that comes from. Each actin can interact with 3 myosins. Here's an actin. 1, 2, 3. So there's lots of interactions that occur. Remember, this is happening in 3D space. Now, the good news is for most muscles, it's linear, right? So when my bicep is relaxed, it's hanging here. When it contracts, it's a line, right? You can think about a line that goes all the way up. For skeletal muscle. But for smooth muscle, around your intestines, around your arteries, that's not true. There isn't a line. And around your stomach, it's even less of a line, right? Because the stomach contracts in multiple directions at one time. And so this relationship doesn't work. This system doesn't work for smooth muscle. Can't. It's too linear. And those muscles don't work in a linear fashion. They can, but they have to do other directions too. So if you did this is an electron micrograph of what a sarcomere would look like. So don't worry about that. I'm not going to have you identify it. This shows you what would happen. Here are 2 sarcomeres in the relaxed state. Here's what they look like in the contracted state. What ends up happening here is you lose, right, because it gets smaller, you lose most of the gap between the myosin and the Z disc because you compress tighten. And you lose most of the gap between actin and actin here. Okay? If you've ever jammed a muscle or had it lock up, you can actually over-contract this thing so that it all gets jammed up temporarily, and then it doesn't want to unlock very easily. You can jam this whole system up, right? This is normal length, so that you've got an actin molecule jamming into another actin molecule. You don't want to do that, right? But under normal circumstances, the 2 ends of the actin here don't touch, but the gap, the H zone, becomes much smaller. And then on the reverse, it looks like this. Relaxed, contracted. So this is just another way of showing this, right? And so you have these golf ball globular heads, right? During contraction, when the globular heads connect to the actin, that is called a cross bridge. The sliding filament theory is based on the fact that you form a cross bridge. Now, here's what gets confusing. The actual head itself, the dual head, they're also called cross bridges, right? They have their light chain myosin, but they're also called cross bridges. So the cross bridges form cross bridges with the actin. It's a little confusing. I don't like the terminology. I like to think of it simply that when the 2, myosin and actin, come together, they form a cross bridge. And it is that cross bridge formation that causes a contraction. It's like the gun got cocked, right? As soon as these touch, you have a contraction. Question. Hopefully somebody's been paying attention. What is preventing these 2 from touching? Alex. The other one. Tropomyosin. So because of the tropomyosin, it doesn't happen. So the question is, how do you get the tropomyosin out of the way? Right? Because if you can't move the tropomyosin, you actually have a different problem. If you don't move the tropomyosin, what's your problem? You can't contract. You can't have contraction. You can't. If they don't touch, this reaction does not happen. And the tropomyosin is a physical barrier. That's what it does, but only in skeletal muscle and cardiac. Skeletal muscle and cardiac muscle are very similar. Smooth muscle is really the strange beast. So once you understand skeletal muscle, taking that step to cardiac is a little bit of a step. You're like, yeah, I get it. They're not that different. But when you go to the smooth muscle, you're like, oh, wait a second, this is just weird. And it is, it's just different. Okay. I don't know if this will actually work, because it never does. But let me try. No, it won't work, sorry. It never does, but it's supposed to. It's like a lot of things. All right. All right. So what actually happens? So this sheet that I gave you with excitation, contraction, coupling will actually explain. We put this nervous system together with this, and we could start with the one on the far left, but this sort of takes you through. Okay, and I'll show you this, but you have it in order, in sequence for yourself to look at, right? And so. Let's just stick with assuming that we're already at the neuromuscular junction. We're already started this contraction process because we're skipping a ton of steps there. All right. They're separated when the two are not touching and it's not contracting, right? This binding site here, okay, this binding site here when it's cocked, when it's ready to go, binds ADP. Right, adenosine diphosphate. That's what's bound to it. And the head is cocked back like this. So when it's cocked back like this, remember their heads from both sides, ADP is bound. Okay? When calcium binds to troponin I, it causes a conformational change. Right? It causes a conformational change that pulls the tropomyosin away from the binding sites here. Okay? So the blue rope, the blue string, gets moved out off of the binding site on G-actin. You can see a little one there. When that happens, immediately these globular heads are attracted to those binding sites. If the calcium does not come in and bind to the troponin molecule, which then causes a conformational change, the tropomyosin stays over the binding sites, the tube, the myosin head, and the actin cannot bind. Okay. Everybody understand that? Is that clear? Okay. Now, once you get a connection, a cross bridge, you get a ratchet, the sliding filament. So the filaments will slide by one another. Now, in order to stop the contraction, ATP has to come in where ADP was and bind to that site. So an ATP molecule will kick off an ADP molecule. As soon as that happens, when ATP is bound, these globular heads no longer have an attraction to actin molecule. So that's step one. ATP binding to this head causes the two to separate. Now what has to happen is you have to hydrolyze, which means you have to cleave a bond, okay, between the outermost phosphate, okay, and this then gets converted to ADP, and that cocks the head again. So whenever you see ADP bound, you know that the muscle is ready to contract. If ATP is bound, that means that the actin and the myosin are, one, not connected, and two, this thing is not ready to contract again. So that's what it says here. The full hydrolysis and departure of ADP plus PI causes the flexing of the bound cross bridge. I don't like that terminology. I don't like what that says, so I give you two of mine. All right. I think that's enough for today. Questions? I would start looking at this sheet, excitation, contraction, coupling. Now this starts, you already have a lot of background on the nervous system. This is going to start to put the pieces together. I will take you through next time, in part of the class, I will take you through the very left column to show you and explain to you. And then it's going to be up to you to then say, okay, I can learn the middle one, because the middle one, there's just a few different things. And then we can talk about the far one at a later date. But the middle one isn't that different. If you see, there's a lot of overlap. Okay, but we'll go through one. I'll go through the easiest one with you to explain it. You guys can ask questions. So I would say start looking at this one. For next time, we can definitely talk about it if you already have questions. All right, three most important things. Three most important things.
Know the hierarchy muscle.
Hierarchy of muscle. Number 2. Everybody wants to leave. Study the sheet. Memorize. Number 3. Sliding filament theory. Yes. Go 4. Do good science. All right. Yes. I just have to